Hello everyone, this is Dr Bishal Gyawali from Queen's University, Kingston, Canada. We are continuing with our Skills Lab series in Medscape. So you must have watched our previous videos. We talked about abstract, and in the last video we talked about introduction, and we started with methods. And the first section of the methods is always about the eligibility criteria and the patient population, whether it's representative of your real world patients. And we talked about randomization. Today we'll continue with the methods section, and we'll talk about the control arm, because the next section in methods of after eligibility criteria is what interventions are given to the experimental arm and to the control arm. And that's why this section is quite important. First, to know what the dose and the schedule of the treatment is for the interventional arm, which is a new treatment, but also to see whether the control arm is ideally what you would offer to your patients if not for the trial. So the fundamental principle of a clinical trial, a phase 3 clinical trial, is to test whether the new drug is better than the standard of care. And the standard of care should be represented in the control arm of the trial. But we have seen several problems with control arm with control arm of the trials in modern clinical trials. So the first question when you are reading the method section and you are reading about the control treatment, you need to ask yourself is this control arm appropriate? Is this control arm what I would offer to my patients if not for the clinical trial? And the control arm may not be appropriate. And how do you decide that? There are three ways in which the control arm can be gamed, so to speak. First, using a control arm that has already been proven to be inferior in the previous trials. Second, not allowing the physician to use an effective drug. This typically is written as the control arm was the treatment of physician choice, and then they will exclude certain drugs that you would ideally want to use. And third, using placebo or ineffective drug instead of an active or effective comparator. And in fact, one of the studies has shown that 17% of FDA-approved drugs have actually been approved based on trials using an inferior control. So I'll give you an example. So first, category that I talked was using a drug that has already been proven to be inferior in the previous trials. So this trial of cemiplimab in patients with non-small cell lung cancer first-line PD-L1 of 50% or more used a control arm of chemotherapy. So this trial ran from 2017 to 2020. For this group of patients, we already knew pembrolizumab was superior to chemo. It improved PFS, it improved OS, and it received approval in 2016. But this trial of a new drug ran from 2017 until 2020. I can't imagine any patients who would not have problem with accessing pembrolizumab, not getting any immunotherapy, and getting chemotherapy. But this trial used a control arm of chemo and received approval based on that trial. So this is clearly an example of a bad control. The fundamental principle is no patient should be harmed as a result of participation in clinical trial. We can make an argument that patients who got randomized to the control arm of this trial were harmed because they did not get immunotherapy and got chemo. One more example. This is a trial in melanoma testing binimetinib versus dacarbazine. This trial ran from 2013 to 2015. And we know that even before this trial ran, and of course until the trial ended, we already knew ipilimumab and then nivolumab should be the standard of care for metastatic melanoma. But this trial used a control arm of dacarbazine, which is unthinkable. So that is an example of using an inferior control. The next category is not allowing to use an effective drug. I'll give you an example of this. This is the trial of olaparib in metastatic breast cancer patients with BRCA mutation. And the methods section will say patients were randomly assigned in 2:1 ratio. So now I think you need to be careful about this. We talked about unequal randomization in our last video. Instead of 1:1, they're randomizing 2:1. And I want you to recall what are the issues related with unequal randomization. Anyways, we are talking about control arm today. So patients were randomly assigned in a 2:1 ratio to receive either olaparib or standard therapy, which is chemo of physician's choice. Now, if you read this much, you would not find much problem with the control arm. It says standard chemo of physician's choice. But then if you go into details about what the physician's choice is, it is actually not any choice. The choice is limited to 3 options of capecitabine, eribulin, or vinorelbine. For patients with BRCA mutation, I would assume physicians might have wanted to use platinum agent because it is shown to be effective and better. But the language does not say we are not allowing to use platinum. The language says we are allowing physician's choice, but the choice is between these 3 drugs, which does not include platinum. So effectively, they are preventing platinum from being used. And the third category of a substandard control is using a placebo control instead of using an active control. The example is the POLO trial in pancreatic cancer. And as you may recall, in POLO trial, patients with metastatic pancreatic cancer first line, they received FOLFIRINOX for 4 months. And if they were responding, they got randomized to get olaparib or placebo. Now, in the real world, when we are treating patients with pancreatic cancer is a lethal disease. And if patients are responding to chemo, we become happy and we continue to use the chemo because it is responding. If some patients have problem with neurotoxicity or other issues, we might want to remove oxaliplatin or one of those drugs, but we'll continue with the rest of the chemo. I can't imagine a scenario where we would start chemo on a patient with pancreatic cancer and confirm that the treatment is working and then just stop it after 4 months. We will not do that. But in this trial, they stop treatment after 4 months and then randomize patients to get olaparib or placebo, which is definitely not the standard of care. We would not want to put them on placebo. But that's what happened in this trial. And then PFS was improved. Of course, it would improve because these patients in the placebo arm got nothing. But even then, overall survival was not improved. In our subsequent videos, we'll talk about surrogate endpoints and overall survival. But just to give you an example of how our trial landscape has become, PFS improved when the patients did not get any treatment in the control arm. But even then, OS was not improved. And quality of life was reported as maintained, which also means it was not improved. But just to, we will talk about quality of life and surrogate endpoints in detail in our future videos. But just to give you an example, improving PFS against placebo, but not being able to improve overall survival against placebo, this, in my opinion, does not count as an effective drug. And these are not, I'm not picking any specific examples. This has become quite common nowadays. There was a trial published just last month in patients with metastatic hormone-sensitive prostate cancer, the phase 3 ARANOTE trial of darolutamide versus plus ATD, androgen deprivation therapy, versus placebo plus ADD, androgen deprivation therapy. This is quite concerning because we have several trials by now that has replaced ADT alone as the standard of care in the control arm. It's not the standard of care. The standard of care for these patients is ADT plus, it could be docetaxel based on the charted trial, it could be abiraterone based on latitude, it could be enzalutamide based on Enzamed, it could be apalutamide based on Titan trial. But ADT alone is not the standard of care, cannot be the standard of care. But this trial used ADT alone as the control arm. And again, I would argue that the patients in the control arm were clearly harmed as a result of participation in this trial. And sometimes people argue that, okay, these trials were run in low-income countries. But if you look at the trial, the countries that participate in this trial, there are countries like Canada, Australia, New Zealand, Europe, which are not low-income countries. Again, these are not isolated examples. We have seen a systematic problem with bad control arms in several modern clinical trials. I would encourage you to read these papers where we have highlighted about this issue with control arm. Who is going to control the control arm? And just to toss on that specific argument that some people make, saying that, okay, this trial was conducted in low-income countries that would not, and this control arm was appropriate in those countries. So can access to the new drug in LMIC justify conducting a trial that would not have been considered ethical in high-income countries? And the answer actually is no, for two reasons. One is the Helsinki Declaration, which says we have to use the best standard of care and not the local standard of care. And second is there is no guarantee for continued access to this treatment after the trial ends. The whole purpose of this trial was to get a trial done that would not have been ethical in high-income countries, but complete the trial and get the drug approved in high-income countries. And patients in LMICs will continue not to have access to that drug. And this has been studied systematically. In this particular study that I am showing here, they looked at drugs that were that had trials conducted in LMICs and got approved in the FDA by the FDA, and they looked at whether these drugs were available in these countries. Patients had access to these drugs in the participating countries. And within 1 year, 0% of LMICs had access to these drugs. Within 5 years, it was 22%. If you look at the graph, then if you look at the graph of Africa, this is quite telling. Up to 5 years after the FDA approval, 0% of these drugs were available in Africa. That means patients from Africa participated in these trials, got the drug approved by the FDA, but even 5 years later, none of these drugs are available in Africa. So we have written a number of papers about this, the challenges of globalization of cancer drug trials. And again, I would encourage you to read this. But just to end today on placebo-controlled trials, sometimes people think that using placebo automatically means that the trial is bad, which is not the case. If the standard of care is an active treatment, there should be an active treatment, but just using placebo automatically does not make the trial bad. A placebo control is justified, and in fact, it is important in two cases. One, if we are testing a combination therapy. For example, if we are testing drug A plus drug B versus drug A alone, then it is better to do A plus B versus A plus placebo. And second, when the standard of treatment actually is no treatment or observation or best supportive care alone, then it can be a trial of a new drug versus placebo. So that ends our discussion today on control arm. On our next video, we'll touch on more issues with the methods. Thank you.